Now, after these two success stories of the TPA project, we're now moving on to the Translation Manchester Research Network Partner Lightning Talks. So this is be like a quick fire version of talks. So all the speakers will introduce themselves and just come up after each other. If I can ask the speakers to either stay close to the podium or to take one of the roaming microphones down there, just so that we can hear you even if you're walking over to the slides. So if we can advance to the first speaker, maybe, so we know who's the first one. And then I just ask the first speaker to come on stage.
Good morning, everyone. So my name is Dami Olanje. I'm the principal scientist for Manchester Biogel. So today I'll be talking on animal-free tunable peptide hydrogens for cell culture. So I would like to believe everyone in the room is probably used to the concept of cell culture, and with the advancement from 2D into 3D, towards more of the complex cell culture models, it's really important to have, the right hydrogens for your intended application. So as a company, we thought I'll probably just share the background on scaffold. A lot of people have probably used different scaffold for different applications, but when you look at biomaterial scaffold, broadly can be broken down into natural or synthetic. So under the natural ones, you have things like polysaccharide, proteins as well. It's got advantages in the sense that they are biocompatible and they are functional. On the other hand, it's got a lot of limitations, things like batch-to-batch variability, not defined or scalable, not translatable, and also issues with strength. So a big player within this aspect is things like Matrigel, which a lot of us may probably be used to the material. On the other hand, we have things like synthetic, where we have metal, ceramics, polymers. They also have advantages in the sense that they're reproducible, fully defined, scalable, cost-effective, and ethically compliant. On the other hand, they have also limited issues in terms of limited biocompatibility. So as a company, what we've done is to pull the advantages of a natural scaffold and synthetic scaffold into what we call peptide hydrogens, and that's making use of the natural building blocks of any human tissues. So we market the products as peptigels, and it's got a lot of USPs in the sense that it's animal-free, reproducible, biocompatible, ready-to-use, transparent, printable, injectable, and sprayable, and also because they are fully synthetic, it's got that translational relevance to its use. Broadly, since the start of the company, we've really expanded the use of these hydrogens across different applications, ranging from complex models like spheroids, organoids, drug discovery, liquid handling for IEEE application, disease modeling, bioprinting, and also regenerative medicine for cell therapeutic application. So if you'd like to know more, that's my email on the screen, and I've also got a stand by table 14. I'll be happy to speak to you. Thank you.
Hi there. So I'm Mohammed Jaber. I represent the University of Manchester as a sponsor, and what we specifically do is work on drug and device trials. Now, if you're translating research, at some point, if you're intending to have it trialed within patients, you're very likely going to come to our office. And the purpose of your trial will always be around safety, if it's an early phase study, and also looking at efficacy. So this is not just drug trials, but it could also be medical device studies, it could be physical devices, it could be artificial intelligence, and likewise with drug trials, it could be something that you're repurposing, which is already marketed, or it could be an advanced therapy. And we have experience of all of that. So just to underline the point, sponsorship is a legal responsibility that the university undertakes. And it's, so as I said, it covers finance, indemnity, and management of the trial. And the reason it's highlighted there in red is it's not just ethics or red tape, is because it isn't that. And it's important to convey that message right at the outset. Because we're fundamentally responsible for demonstrating compliance with what's called good clinical practice, with how a trial will be conducted, but also, for example, if you're using any drugs that are being manufactured, we've got to ensure the quality of those drugs has been manufactured to GMP standards. And there are various other standards that we would need to adhere to. We're regulated by the MHRA, which is a Medicines Health Regulatory Agency, but we also have exposure to the European Medicines Agency and the FDA. So this is just to give you an idea. If you were thinking of setting up a drug trial and getting it from start to finish, the bits in red are just law within the UK that you would have to adhere to. So it's not as straightforward as one may expect, thinking we've got everything, it's ready to go into patients, can we just run it? It is a bit more convoluted, it is a bit more complex, and it is based on risk. So we have a team here within Manchester. As I said, we've built up over the past 10 years. We've worked a lot with academics, clinical academics as well, and building up our experience and insight in not only setting up drug and device trials, but also looking at what the risks are with trials not completing to time and target. I mean, a golden rule of clinical trials is they're never repeated. So it's important to get them right from the outset. So making sure they're costed appropriately, you've got sufficient time to set them up, and there's enough time to see that trial through to completion and archiving. We work very closely, because we're within the Directorate of Business Engagement, we work very closely with the contracts team. And as it's important to point out, contracts don't just deal with the financial agreements. If you're working with vendors that are manufacturing products, providing any kind of management for your trial in any way, shape or form, all of that would have to be overseen and would come under the ambit of a sponsorship. We would have to be assured that these organizations can fulfill the requirements for our trials. As I said, we work very closely with research support managers, ensuring that the costs are there. We're tied in with business engagement and also insurance. Indemnity is actually a big thing for trials. It's something just to note. Whilst the university has fairly comprehensive cover around clinical trials, there are some exclusions. So depending on what you're working on, it's always worth just reaching out to us just to get that clarified as well. And we're also heavily networked with other organisations in the UK, especially other Russell Group universities, and likewise with our partner trusts in and around Manchester as well. And we also work quite closely with the regulators as well. So as I said, we've got lots of experience now with a variety of different drug types of trials, devices, and also if you're, for example, involved in developing diagnostics as well. So we have some experience around that. Just to let you know, just focusing on devices, we've got experience of self-certifying a device. So there are different types of devices. We've also led on class 3 devices, which are the highest risk. So surgically implanted devices, for example, we've not only been sponsor of those devices, but we've also been the manufacturer of those devices. And that doesn't mean we just physically put it together. It's the fact that we as an organization own the know-how around that. So it's important just to note that. And then just to finish off, if you are working on any of these kind of ideas, it doesn't matter how early it is, it's worth reaching out to us at this stage so we can actually work with you and start looking at how to basically get your product into the clinic. And there are a couple of email addresses where you can reach out to us. Thank you very much.
Okay, I'm John Waterton, University of Manchester and Bioxidine. Bioxidine was spun out 2009. It was right in the middle of Luke's slide of University of Manchester spin-outs. So next slide. So I'm here representing imaging biomarkers and representing Bioxidine. There's quite a lot of people here today representing imaging biomarkers, including our chair. So I want to speak firstly about that concept and then tell you how Bioxidine can help you. Imaging biomarkers have got a lot of advantages. They're great for focal disease, unlike circulating biomarkers, which integrate over the whole body. They're generally non-invasive. You can follow up as often as you like. But most importantly, they have exactly the same regulatory status as any other biomarker modality, circulating biomarkers, IHC, that you may be more familiar with. And if you're not familiar with the best resource put together by NIH and FDA in thinking about clinical trials, you should be. But there is a little wrinkle that the standardization and validation roadmap is very different for imaging biomarkers than biospecimen biomarkers. A lot of the tasks we do are not incentivized in academia, and CROs spin-outs have a major role here. There is a roadmap which was put together and has been very influential, led by a number of us here at the University of Manchester, but international in scope, that lays out some of the activities you need to do that are very different from the more familiar molecular biomarkers. So what does Bioxidine do? We support imaging biomarkers, particularly MRI biomarkers, in a whole range of important therapy areas: inflammation, lung, arthritis, cardiovascular, liver, kidney, neuroscience, and very importantly, oncology. Please come along to our booth where we have a lot of fabulous examples of how we've helped both academic and pharma companies deliver imaging biomarkers for their trials, including, of course, a number of studies with the University of Manchester. What can we do for you? So firstly, most importantly, we can advise you on what imaging biomarkers you might want to think about in your clinical trials, including perhaps things you haven't thought about before. We can help justify imaging in your grant proposals or clinical development plans. And then we can help you deliver them, setting up sites, qualifying sites, working to GCP 21 CFR 11, fully compliant all the way through from phase 1 to phase 3 and beyond. And we can help you with supporting you with publications and regulatory submissions. So I hope I kept time.
Good morning. Hi everyone, my name is Niels Peek. I work in the School of Health Sciences and I'm also director of the Christopher Pankers Institute for Health Technology Research and Innovation. The institute was already mentioned a couple of times this morning in Luke's opening speech and also by Graham later on. So as I mentioned, we were established last year as a virtual institute, and soon we will also have our own building on Dover Street, right at the heart of the university campus. I think it's really important that we're going to have that institute because it will enable us to enable people to work together across the boundaries of their disciplines. So we're going to have a number of people who are going to be based permanently there in that building, our own staff, the Translation Manchester staff as well. But we're also going to have hot desking facilities and meeting rooms, because I think in this space of health innovation, and health technology translation, is extremely important to work across the boundaries and help people who come from the technology area to understand, for instance, what it takes to bring things into frontline health and care practice. It's important to mention also, as the slide tells you, that we are not just University of Manchester, we are a partnership with other regional stakeholders in Health Innovation Manchester. So we're part of the Manchester Health Innovation ecosystem, and our funding partners are Manchester University NHS Foundation Trust, Manchester Science Partnerships, which links into the technology and life sciences industries, GMCA, Combined Authority for the region, and Health Innovation Manchester. In terms of what we do, so this slide tells you what our activities are. We've got three broad technology areas represented by the pillars here. So digital technologies, materials, and AI. And then we have a number of cross-cutting themes. And some of those themes are very well established, and others are more emerging. I'd like to mention the work that Robert Mackin from the School of Social Sciences is doing. So Robert is leading our responsible and inclusive innovation theme, and he's looking at, he's studying the processes of collaboration between different disciplines and processes of translation. And I think that's extremely important. I think we all agree, otherwise you wouldn't be here probably, that translation is extremely important. But I think very often we still need to improve our understanding of what that really is and how translation processes really work. So that's what Robert is doing. Our translation lead is Claudia, who's here, and I've got another slide on the work that she is doing. And what this slide tries to tell you is that we believe very much that translation, this is something that Graham already mentioned as well, is not a linear process. And especially for new technologies, it can be very challenging because the processes of translation innovation are not well established. So for instance, regulators very often struggle with new technologies and say, well, we can't regulate this. This is beyond what we can do, really. So what we believe is that it's important to start thinking about translation innovation from the very start of your research. So it has to be an integral part of health technology research from the very beginning. And what that means is that you have to engage with the relevant stakeholders, and again, from the very beginning. And those conversations will then evolve over time as the technology matures and becomes ready to be trialed and adopted in clinical practice. So we're going to be here all day. We've got a stand as well as the exhibition area, so feel free to approach me or other colleagues from the Pankhurst if you want to know more. Thanks.
Morning, everyone. Thanks, Alicia and Fiona, for asking me to talk briefly about the Manchester Renal Biobank. I'm Durga Kanigachel. I'm one of the nephrologists and transplant physicians from Manchester Foundation Trust. I'm probably one of the slightly odd one here that I come basically as a clinician into the center here. I'm not going to tell you that kidney disease is the only disease that everyone should be concentrating on, but certainly my life is spent in kidney disease. And Manchester Biobank is one of the few odd ones in the sense that we have a biobank within the Manchester Foundation Trust with the clinical repository. Now, kidney disease is one where patients with involvement progress from one stage to the other, and stage 5 kidney disease is the end stage kidney failure, or people, what we call as kidney failure, needing renal replacement therapy in the form of transplantation or dialysis. Now, that is a hugely expensive one, and unfortunately not so well researched considering the amount of burden it costs both for society, individuals, as well as across the world, shall we say. So the main aim of setting up the biobank is primarily to try and see whether we can come up with strategies to prevent people from getting into kidney failure, although we also can recruit patients after kidney transplantation or dialysis. Now, the commonest cause of kidney failure is diabetes, but that constitutes about 30%. There are numerous other conditions which are considered as rare diseases, and they're, as you can see, 2/3 of the conditions are related to rare diseases. And they're so nuanced that, I think Catherine Ells was saying earlier, that antihelminthic drugs are from 1970s. Actually, we still use treatments from the 1980s and 1970s for some of these kidney diseases. So there's a huge ground to be made. Certainly from patients who come to us in terms of the clinical setting, we cover the east and the south sector of Greater Manchester, which constitutes about 1.5 million population, and about 5 million for transplant populations, both in Greater Manchester as well as in Lancashire and South Cumbria. The primary aim for setting up a biobank a few years ago was just to develop a resource for kidney disease research locally, and the advantage of this is because we have a circumscribed population and you can do some potentially epidemiological research as well. And the tenets are to, of course, increase the precision medicine through innovation, and both from diagnostics as well as from prognostic perspective. The repository has, because of the way the IT in the hospital in MRI is set up, we have a clinical repository which is entirely IT-based, so we don't have to go through any paper notes, etc. And of course, we have the repository in terms of blood, urine, and most importantly, kidney tissue in both either a fresh kidney tissue or in the frozen form. And it's REC approved from the Northwest REC Committee, sits under the MFT R&I division, and we started this back in 2016, although there are previous versions which actually we still have samples for patients going back to the 1980s and 1990s. And there have been a few research publications which have come out of it. The aim is to get up to 1,000 patients by the end of 2026, which is a bit ambitious considering that these are rare diseases, but we set ourselves a target. and we can include both adults and children into the biobank. Recruitment is during any part of the natural history of kidney disease and at the time of renal replacement therapy. We also have healthy volunteers who can participate in the study itself. Ultimately, of course, it is to generate more data from a genetics or markers from a biomarker perspective, both from a proteomic and metabolomic perspective. The unique nature of this biobank compared to any other biobanks that you come across is there is a potential for, or there is ethical approval for prospective collection, and therefore you can have some serial samples if you want to study the state of the disease and the biomarkers, etc. Of course, not necessarily kidney biopsy. We don't repeat kidney biopsies, but we do have sample collection prospectively. And I'll stop at that. Our contact details, Patrick, Omar, and myself from the adult division, and Rachel Lennon from the pediatric division. Thank you.
Morning, everyone. So my name is Faith, and I will be presenting on behalf of the Complex Wounds Biobank Manchester. We are a team of biomedical researchers who are based at Wythenshawe Hospital, and we also work cross-site at Manchester Oxford Road and within the university as well. So today I'm going to be talking to you about the types of samples that we take and how we can help you with your studies, and also just touching on the types of applications that we accept and how we can also help you provide extra information for the data that you need. So to begin with, we collect a wide range of samples. Most of them are from discarded spare surgical material, but we also take blood samples, wound biopsies, dressing samples, and we can also take, depending on your study or depending on your application, fat and fascia samples. That is currently something we're doing at the minute. Our most common is wound tissue and biopsies, but we do also have availability to a different variety. For instance, we can take extra skin tissue, or we can take extra, like I said, fat tissues, or extra serum and blood samples as well. It is all study dependent, but we're not limited to a specific set of samples. We would go through our ethics advisory group and assess what you need and assess if it's compatible with our team and our facilities at Wythenshawe as well. So the study management, as I said, we currently take a lot of discarded samples or samples that would be discarded. Now these samples tend to be either requested fresh, delivered to the end user, but we also can store them or freeze them depending on what your requirements are for your studies. We do also tend to take quite a lot of dressing samples. Now these would be from our clinics, provided by our clinical team and our burns and plastics nursing teams. These would be most likely delivered on the day after they've been taken. But we can also, depending on the study, again, figure out, depending on storage and use and requirements, figure out how to store them or how to extract what you need from those samples and get them to you. So all our samples do go through our ethics advisory group, and mostly that would be our clinical trials coordinator and our PIs as well. They would assess whether we have the capacity within our admin and clinical team, whether we can also access the data that you need, and how we can distribute that data to you. There is also an element of financial needs and also any, for instance, future transport needs and future career needs that you would need as well from us. If you would like some more information, please do head over to our store. I've got a lot more to tell you about us. We've got stickers, we've got sweets, and we've also got some pens and lanyards as well. We are on store number 31 in the Pioneer Room. Thank you so much.
Hi, so I'm Darren Green. I'm the clinical lead for NCARC. NCARC is an HTA licensed tissue bank and biorepository which is hosted by the Northern Care Alliance So it covers the 4 hospitals housed within the northern parts of Greater Manchester. NCARC was first established in 2018, and since then we've had nearly 60 applications for access to our tissue. So we do supply stored tissue, but we have a very flexible approach where we can provide bespoke collections for specific projects, and that includes fresh tissue. So as an example, we have an acute coronary collection that supplied more than 600 fresh tissue samples to an external partner in little over 12 months, and that comes with a very, very highly granular level of clinical data from our electronic patient records. So NCARC does have the ability to provide very, very bespoke service, and we have a quick turnaround from when people approach us to when we can either release samples or begin a new collection. So these are our currently active collections. We put them into 4 domains. Our most recent collection that started is a rare and ultra-rare diseases collection housed within the metabolic medicine team at Salford Royal, and that sits within our cardio-renal metabolic area, which has been very fruitful in developing new partnerships between the Northern Carolines and the University of Manchester for some early stage translational studies. We have a number of surgical collections across GI and particularly neurosurgical teams. The neurosciences domain has a lot of collections specific to different disease areas, as you can see there. Our area where we probably have the most success in terms of grant outcome, research papers and cross-pollination between different clinical areas and different mask partners is the respiratory collection. So you can see there we have quite a number of types of tissue there, so pleural samples, pleural fluid, swabs of various origins, COVID samples, lung cancer samples, and again these are either stored or can be taken fresh. And within this we've had, say, a number of grant applications that have been successful. One of our most recent fruitful collaborations across MASK has been a collaboration with MMU, microbiology team and the cardio-renal team looking at lung microbiota patterns in patients with long-term pulmonary edema. I say what underpins this is we have a very engaged team and a team of local experts where we can hopefully provide very bespoke service for what people need within their research studies. This is our contact details, and again, as with everyone else, we have a stand in the back room.